Here insulin hotly regulating the gene expression. As we know, insulin generally in vastly we know the insulin actually regulates the carbohydrate metabolism, right? So for carbohydrate metabolism, as we know, there is a different type of an enzymes is also required, and the functional proteins transporter everything is required for the carbohydrate metabolism. The carbohydrate must has to come inside the cell, so for that we need a transporter. So like that, different type of an enzymes and the proteins are required to complete the process of the carbohydrate metabolism. These proteins and the enzymes came from the gene that is present in the nucleus, right? So to increase their number, insulin give the signal to the cell that there is a huge amount of a glucose or pretty much amount of a glucose in the blood. So we have to process that glucose. To process that glucose, we need to synthesize more proteins and the transporter. So that's why insulin give the signal to the cell through the transmembrane receptors. Signal will come inside the nucleus. So how it will start? It start when the insulin bind with the membrane receptors. When insulin bind with these membrane receptors, the membrane receptors get activated. And the insulin receptors is a dimer. It is the dimer, and the dimer have a two component or the two subunit. One is the alpha subunit and another one is the beta subunit. Alpha subunit is this outside of the cytoplasmic membrane or the outside of the cell, and the beta subunit is the inside the cell, or it is the cytosolic side. And another interesting thing is that also have a tyrosine kinase activities. So one subunit transfer the phosphorus group from the ATP to the another subunits. Okay, here we also see, so transfer the another subunits. So in this way they actually phosphorylate the receptors molecule. So when the receptor molecules get phosphorylated, they become a point of nucleation. That means they become a point, docking point, where others protein can bind with the help of this phosphorus group binding sites. So first of these proteins is the insulin receptor substrate 1. This substrate 1 have the binding site for the phosphorus group. The phosphorus group that is present on the receptor sites, right? When it is bind, they used to get phosphorylated, and after phosphorylation of this insulin receptor substrates 1, they again this one become a nucleation point. So this SH group become phosphorylated, and since it is become a point of nucleation for the other proteins, so first protein who will bind, it will bind with the GRB2. So GRB2 have a two binding site or the domain. One is the SH2 domain and another one is the SH3 domain. With the SH2 domain, they bind with the phosphorylated. Now GRB2 have the another domain, that the SH3 domain. With the SH3 domain, they bind with the SOS protein. This SOS protein exchange the GTP of the RAS proteins. Okay, so RAS proteins actually exchange their GTP with the GTP. When they exchange the GTP, now this RAS protein is get activated and bind with the RAF1 proteins. This RAF1 proteins phosphorylate the MEK proteins. When the MEK protein get phosphorylated, these MEK proteins get activated, right? This activated MEK protein now phosphorylate the ERK proteins. So these ERK proteins get activated. So these activated ERK proteins now from the cytoplasm go to the nucleus. Okay, so in the nucleus activate the ELK1 protein. ELK1 protein gets phosphorylated. Now ELK protein with the SRF protein get dimerized and they bind with this DNA molecule and they function as a transcription factor. These transcription factors now start transcription. So this pathway is active unless the requirement of the protein is fulfilled inside the cell. So this is a cyclic process, and at the same time it is a cascade process. Cascade in the sense one molecule activate the another molecule, that molecule activate the another 10 activate the another 100,000, like this signal get amplified. So this is the complete pathway of the insulin hormones signaling. That means how insulin hormone regulate the transcription of the genes that is required for carbohydrate metabolism.